Okay, there was a question from last time on Piazza about some clarification. So the question was that while we did the example, why did we take a particular route and not some other route? So the short answer for that question is, if you have clarification, if you still require clarification, sorry, this should say, I think, it's lecture 7. Okay, yes, that's correct. So there's is it lecture 7 or is it lecture 6? Lecture 6, yes, sorry, I'll correct that. So the clarification that was asked for was why did we take a particular route? So when we take the choice, right? When we are making a choice in global alignment, we make a choice for the most optimal score that we get here, and we store it. That's the thing that we store to figure out where did we come from until we reach this step. And once we reach this step, we backtrack. And that's why the path that you get is the path that you see towards the beginning might not be the same path that you take backwards. This will become only clear if you go and refer to that Piazza post or you do the assignment. But if that my response is not very clear, that's I just gave a hint that you look at the backtracking path and not the path that you come. That's that should hopefully help clarify the question. Any questions from the last class or anything that was not clear besides this point? This came on Piazza, which I just answered very briefly. Okay, so if not, then we'll begin. We will not have a class on Monday. There's a guest lecture in BSB from Professor Ullas. He will talk about some other stuff, not purely things that we do in the class. But I think given there is some overlap, I thought it would be useful to attend that lecture anyway. It's at 4 p.m. in the BSB building. I will strongly encourage you to come. If you come, there's a bonus points of +1. There will be an attendance or a sign-up sheet. We will not have a regular class at 3:30. Okay. Okay. So we'll go back to local alignment. We'll take it slow today because I thought I'm trying to pack a lot of stuff in the lectures, so I'm going to slow down a bit. And I think we should still be able to cover it. So if we finish early, we'll start. So now we took a roller coaster, right? So we climbed all the way up. We're slightly going down now, and we will plateau for a bit, and then we'll start climbing up again. So this is that stage where it's easy to see from the top what now we will be seeing. So the first problem that we left last time was this idea of local alignment. And the way to think about this is if you're given a particular sequence from any particular organism, it's not necessary that that entire sequence should map to full-on gene from some other organism. So this could be gene in human, and I could take a sequence which would be smaller. In our global alignment, what we try to do is we literally try to map them end to end. This is what is GA or global alignment. However, it's kind of understood in biology that not all parts of it should align to that gene. Maybe, for example, in the case of that virus, it could just be a very small sequence somewhere in the gene which needs to map, which gives it the function that is required. And that is where the idea of local alignment comes. So this is LA. So local alignment. Rather than forcing the same sequence to align to end to the other gene, we look for the best possible position in the first sequence, usually the larger sequence, where the alignment will be the best. Right, so we already have seen a scoring strategy. We're not technically doing anything different. We're still aligning sequences from one to the other. Any ideas how or what change would you do in the scoring mechanism to align these sequences? The scoring mechanism was given by, if you're aligning the ij base, you can come from, you can align till the sequence and align this, or you can incur a deletion in the second sequence or a deletion in the first sequence. And the way we align this, or the way we initialize this, is we said that we want to really discourage gaps. Gaps are basically dashes. We don't want them, like they're generally not good. So the way we align this matrix is, or we'll just take a very simple sequence. We discourage it by saying that any sort of alignment any sort of alignment will incur a penalty if you start from a dash. Dashes are not good. If you start from dashes, they are not good. Right? That's what essentially this means. That if I start from here and align this, I incur a penalty of -2. Why does that minus initialization help? It helps because we are in essence trying to maximize the overall score, but in the process we are saying that since I want to align, I have to traverse this entire path. I have to traverse this entire path in global alignment. I have to go from starting on the sequence to the other of the sequence, and same for this one. Now if you take that analogy, one thing hopefully you will see is, while this is a small sequence, let me cut this off. If I were to do a local alignment, I'm saying that I don't actually have to traverse the path. I can stop here itself. I don't have to go all the way to the end. The start and the end can happen at any location with respect to the first or the longer sequence. Whereas in this global alignment case, you're saying that no, everything has to be aligned end to end. So these scores need to be negative, because if I force sequences to align to a particular dash, that needs to be, or a particular space, that needs to incur a negative penalty. Any ideas about how can we allow sequences to start from anywhere? What change should you do in the scoring function so that even if I have a sequence like, let's take this example so it becomes a bit more concrete. This is my first sequence, F1 and F2. I have a sequence like GC. If you do this approach, you will land up with something. Right? You will land up with one possible alignment could be, of course, this. One possible other alignment could, if I allow for partial mismatches, for example, in this case, this could also be possible that I start from here. There are a lot of possibilities, but we're not actually looking for this. Like, this is going to force it to have the same length as the original sequence. What essentially I'm allowing for is it's okay. It's okay to start from anywhere, and there's no penalty for giving you gaps. Right? You can have anything in those spaces. It's fine. Dashes are fine. They're not as bad as we thought them to be in the global alignment case. How would you incorporate this? In the initialization condition, what change should we do so that even if my sequence starts from here, it's not necessarily considered to be penalized? Yeah.
So once it's started, then after that, if there are gaps,
that is allowed. Yeah, gaps are allowed. It's not that gaps are not allowed. So we will still have penalty, but there's no opening penalty, because
the starting way is not an issue.
starting way is not an issue to have gaps, right? In this case, if I were to align it, if I get something like this, it's okay. Whereas in global alignment, you're kind of forced to not have these kind of starts, because it will have a very huge penalty, right? So this is already a score of -2, -4, -6, and so on. What change should we do in the scoring function so that it is okay with this penalty? So the scoring function, should this change much? Like, is the entire premise of this function changing if I am doing global alignment versus local alignment? Once I know where to start, it's the same problem actually, right? Like, it's not different from what I was doing here. Is it that figuring out that start is where we need help?
Local alignment, in between we can have gaps.
Yeah, you can have gaps.
That will be good.
Okay, so you will allow for gaps?
That is what I'm asking.
Okay, yeah, okay, fine. So you know gaps are allowed. It's not like gaps are not allowed.
Like GCD is there. For
Hmm.
example, if it had been GCC.
Sure, sure. If it is, let's say, GCA T, that's okay. It can have the alignment there would be that GC hyphen T. This is okay. That's allowed. What change should we do to allow? We have to figure out the starting point actually. That is the main thing, like opening or figuring out where changes should happen. That is where the crux lies. Any ideas?
Remove the penalty for dashes at the beginning.
Okay, so what will you remove it to?
I guess theoretically zero if you don't want to pay anything.
Okay, good. So let's say if I change this to 0, right? What essentially I'm doing in this process is I'm saying that whatever your gap penalty is, it's okay to have gaps in the beginning. This particular sequence essentially aligns that what happens if I align all my bases to dashes, and in local alignment I'm saying it's okay if all of you are forced to begin with a lot of dashes, because I know that the alignment is not going to be end to end. It has to start somewhere and it has to end somewhere. And that is what can be achieved if you don't penalize for beginning or opening gaps. Anything else you would like to change, or does that do it? So let's do an example, and we will figure out what is the crux. But essentially, we do not want end-to-end alignment, so the initial penalties can be relaxed, right? So you can make the initial rows and the columns to be 0 because there is no gap penalty there. We are looking for path stretches that will yield high scores over the length of the smaller sequences. So we can discard paths that are low scoring. How do we do this? Does this idea make sense? We are looking for stretches that will yield high scores over the length of the smaller sequences. So while I said a gap penalty is allowed, we are more interested in maximizing the score overall. Right? So if I reach towards a path where I have to indeed introduce a penalty or do a negative score, I would like to not take that path, because I know that going forward from there, I'm going to lose information. Does that idea somewhat make sense? Once you incur a penalty of -2 here, even if there's a match, in that particular path, you are going to incur some loss in the process. This is best seen through an example, so let's do an example about what does this mean. But for that example to show up, I will have to first show you what the scoring strategy is. So the only simple change that is being done is you're relying on max of this. So your initial condition is of course all 0, but you are essentially, by inducing the second condition, which is that if you incur a gap, that path might not be the most optimal path. We'll not do a formal proof of this. The way to see this intuitively is, once you take a path which has one deletion, in the process you've incurred usually a negative score, and you would like to further not continue that path because you know it's in some sense already become suboptimal, because in this path, in this goal, the goal is to do one-to-one alignment to a location. So while you can incur while gaps are allowed, you're not penalizing them as such in the sense. So that's why your minimum score will not never go below 0. The max score that you get here is out of 0, and the all possible scoring strategies that we saw. So let's do an example and it will become hopefully clear. Okay. So we'll take this sequence, and you know what to do next, which is you're going to try to draw this on the board and see one by one how and which path we should take. Maybe I'll do it here. It's a pretty long one. Okay, so the first thing that you do once you get a sequence like this, right? So even before you do the sequence, let's try to guess what the local alignment in this case should look like. What should the local alignment look like? Let's write it down here. What should the local alignment look like? Any patterns that you see there? Okay, CT. Did I write the sequence now? CTCA. Yeah, CTCA. Okay. Okay, CT. Okay, so CTAA, there's some scope here. What about the rest? Doesn't look like a match, right? It doesn't look like a match. But if I could force somehow, this might be one potential mapping, right? So we'll try to see what comes out of it. Okay, let's fill this matrix. And in this case, +2, -1, and -2, right? So +2 for a match, -1, and -2 for an intel. Your S is max of 0. Should be self-explanatory. What? The previous score plus this score, max of 0. Okay, how should I initialize this? 0, 0, 0. Initialization, gaps are okay. No issues with gaps.
So when
Yeah.
we are doing this, but what is the objective?
The objective is to find out between two different sequences, is there a local sequence? Is there a smaller subsequence of it which shows a good alignment? The reason we are interested in this is to find out if there is shared set of sequences which are present in two different organisms. This could be organism 1, this could be organism 2. The reason sometimes only small subset of sequences will match is because this is what is carrying out the important function. So if I take human sequences and if I take DNA sequences, I don't have to worry that gene 1 in human should look exactly similar to gene 2. Between those two genes, what are the common things which I can find out to the best maximum possible, which give me the best possible alignment locally. That tells me, that can help me understand what is the function being carried out overall for the whole gene. Because while the rest part has changed, this part has not changed. this is across organisms. So let me show you an example. Maybe let me try to motivate this by first showing an example. And the way to show this example is to do what's called BLAST. So in BLAST, we'll study what is BLAST in just a bit. But let's say in my entire in my lab, I come up with a new sequence which looks like my name, for example, right? This is protein BLAST. I come up with a amino acid sequence which is, for example, my name, and I'm interested in understanding what is its function. One way to figure out its function is, does it happen to be present in any other organism? Because if it's present in any other organism, somebody would have characterized this is a very small sequence, so where does it occur and what function is that particular sequence associated with? So one way to do this is to do what's called BLAST. This is a protein BLAST. So let me do this. It'll take a few minutes, or actually a few seconds. So at the back of it, what is it trying to do? It's trying to run a match. It's doing this kind of pattern matching, trying to figure out at what point do we see something like this come up in terms of its sequences. Now, if I take my entire gene, I don't have to align S. I could have had a sequence which would look like this, for example. There is some base and something, and so I don't have to force it to align in a global sense. If this was my original sequence, this is where the rest of the bases are, which are not A or K. If I treat a good global alignment, I would land up with something like this. Whereas if I had some other sequences, which is where the gene is, the sequence that I'm looking for is embedded in this sequence, the input query that I have is this sequence. And in this process, when I inputted this sequence, if you force global alignment, you will have a different alignment as compared to the local alignment. So I'm here interested in finding out what is that subset of sequences which match. And again, this is slightly simplified because of course there could be bases here as well. But the idea is there a subsequence of it which has the maximum overlap with the query sequence, with the reference sequence. So what I typed in is a query, and what came out are different sequences where it has a match. So for example, the first one is from a bacteria, which has some score, some query cover. So it covers 100% of the sequence, the query that I gave. So this is my query, which is my query sequence that I gave it. It has a sequence score of 100%, and there is some value attached to it, which is called a p-value, which we will see this in just a bit, and the percent identity. Right? So this particular sequence happens to be present in this cluster of bacteria, which means that this particular sequence is conserved in bacteria. It's carrying out some function, and I also know, for example, what is its representative sequence. So I can look, for example, this is my query, and this is what the subject looks like. This is the input that I gave, and where the match happened, that seems to have an exact name sequence match corresponding to it. So somebody would have studied the function of this, right? So somebody, let's say that this is in, I think this organism is ferret. In ferret, this happens to be in a gene called mucin 16. So ideally, what I would have done is I would have taken the mucin 16 entire gene and done a full-on global alignment, and then tried to find it. Whereas this is a very small sequence, it appears as a very small part of that entire mucin 16 gene. And mucin 16 gene, somebody who already studied, somebody would know what the function of mucin 16 gene is. Similarly, there is this putative adenine repeat and fibrinogen. There's some function going on with particular gene. With this query, I am able to putatively tell or tentatively tell that, okay, this sequence I also have presence of, so maybe I'm going this sequence in my organism is also going to carry out a X function, whatever that function has been previously characterized. Okay, is that part clear? Okay then, so back to doing this. So how do we fill in the rest of the matrix. So what is the first step here then? We're still doing the same thing, right? Which is where did I come from? So there are three possible ways. I can come from here, here, here. What should I type here? Say that again. Look at the scoring function. So we will do this exercise by looking at this scoring function, and I will strongly encourage you that you repeat this exercise with by deleting this and, well, not this, by deleting this and seeing what it looks like. Initialize it as if you would initialize in a global alignment case, and then repeat this once you go back home. So you'll get a feeling of what is going to happen. For now, we will just do the local alignment case. What should I write here?
0
0 or 1?
0.
Okay. We'll see where. Should I care about the traceback? I don't care about the traceback because I've not even started actually. Nothing has started yet. Okay, how about here? Okay, here. Okay. Here. Okay. Here. We'll come back to see where the traceback should begin. How about this? 0. Here. Lot of zeros, huh? Oh, sorry, that's correct. So, no, so this is correct. So, it's max of 0. The maximum value that you can always get. There will be never a negative score. This is what a lot of people get wrong. There will never be a negative value in this matrix. And that is because we want to discourage path stretches where the score is even going to go down. The moment you have a -1 or a minus thing, since we are interested in having smaller set of sequences to match, we want to discourage paths where we take a negative route, where we force a gap to happen. We're looking for as close of a match as possible. So while you are allowed to do it, you will still incur a score of 0 and not a negative score. Okay. Okay. How should I fill this? What about this?
That would be 4.
Oh, that's correct. Yes. Max. So this is your G is matched to G, and you go from where to where actually? Yeah, you go from here to here, and that's how you get it. Okay. Here 0. Now. Okay. 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 Sorry. Okay, everyone with me? Which one? This one? This line? This one? Okay. So we started from this sequence, right? So wherever you are, you have to sort of ask where did you come from. So you came from here. You would have done a match, but you have forced a mismatch. You would have incurred a 0 -1, which is not allowed. So max of 0. You could have gone from here to here, which is indel -2, which is not allowed. Similarly, 0 to -2, not allowed. Maximum of 0. Okay. Here, you come from. If you come from here to here, this and this is a match. So that's why you get a score of 2. That's the maximum. If you came from here or here, it will be -2 or -2, which will get converted to 0. So max of 0, 2, -2 is 2. So that's why I get 2 there. Okay. Any other questions? Okay, here here.
Zero.
Okay.
One.
Okay. So here 1 because we are here. I mean, we are here actually. We want to score for this. We can come from here, which means -2, which will get converted to 0. We can come from here, which will again be 2 minus 2, which is 0. But we can also force a mismatch, which is 2 minus 1, which will be 1. Okay? Yep. This one?
In the C, second two.
Second 2.
In the C.
Sure. So you are here already, right? At no, so wherever you are focused, so this is the position that we want the score for. I can come from here, which is I can take a path of starting from GC and then adding a match. So this match will be between a match between C and C, so that's why it could be 2. I could have come from here, but would have forced an indel, so 2 minus 2 would be 0. I could have come from here, where I would have again forced an indel, so this would have been -2, which would get converted to 0. So max of 2, 0 minus 2 is 2. Okay? Any other questions? So I'm not keeping traceback for most of them because they're not required. We'll see how to decode it. Okay, here. Right, 1. How about here? Correct, right? So hopefully now we're getting the feel. Here. Okay, this one. Okay. 0. Correct. Okay. Okay. Almost there. 0 here. Okay. Now here I can go from here to here, which means I'm at 2. I'm forcing indel. So I will have to have what? Max of 0. I could have come from here, which means I'm going to get a match, so 2. And again, of course, 1 minus 2 would be -1, so max of will be 2. Okay? This is the trace back. Okay, here T 2, correct. Here okay. Then good. From where? Diagonal? Actually, both work. Right? Both work in this case. We'll see whether we need it or not. How about this? 0. How about this? Okay, getting there. 0 here.
The first one is at 2. A2.
That's correct, yes. Okay. Next, sorry, my losing the sense of verticals and horizontals. Here, 0, that's correct. You can either come from here, you can either come from here. You will get a 0. You can also actually come from here, which basically means that this is a suboptimal path. Moment you encounter a 0, right, that's a suboptimal path. That's why these trace backs don't matter. Okay, how about here? How about here? That's right. From, okay? Here? That's right. Where? 5. Wow, okay. Okay. 3. Is it correct? Yeah, that's correct. How about this? Actually, it doesn't matter, but what? Oh, 1. Okay. You see something arising, right, in terms of what is happening, for example, in this stretch. Your TCA is starting to take some shape. Let's finish the rest of it very quickly. So this will be how much? 2. Okay. How much? 1. Okay. Correct. Correct.
3.
Correct. 6. Yeah. Okay. And 4. Right. 4. That's correct. Yes. Okay. Mostly I don't care actually. That matters. Okay. T. 0. 1. So how did 1 come about? Snatch. Okay. Here. Okay, doesn't matter. Yeah. Okay. Now 2. Okay. Here. Okay. Here. That's right. Here. Okay. How? I did a mistake, is it? Hold on. Just 1 second. Okay, that's correct. So we are at T, and then you are at G. 6 minus 1. That's correct. 5. Okay. Now. Yeah. How? Yes. Okay. You see some pattern arising in some sense. Here you start from your. Let's finish this because we have to take the smaller sequence. We have to go towards the end of the smaller sequence, right? So we can only stop till we reach there. So 2 more to go. We'll finish this quickly. Okay. How much? Yeah. Yes. Correct. Okay. How much? Yes, that's correct. 0. Correct. That's a mismatch. Correct. Okay. Here. Here. C and this is G. Okay. From there. Okay. Final one. Two. Oh yes, that's right. 2. Okay. Zero. Right. 2 is correct. Okay, now. Okay. Okay. 1. No loop. Okay, long journey. Very long journey. You will do this once in your exam, but otherwise you will use computers, right? Like this is still a good exercise to have. The reason to do this is to get a sense of like at what point, what is it that you're doing. The reason to begin with the zeros is so that you allow for indels. The reason to not care so much about a deletion is because that's a suboptimal path in some setting. Okay, how do I trace back? So I still have to take the smaller sequence now, right? So I take the smaller sequence, so I still have to go towards where I landed up with and then trace back from there. Okay, so I'm going to trace back. You can see there's a lot more diagonal elements, right? There's not much, except in this case, there's not much off diagonal paths that we took in that sense. I'll give you the solution, so don't worry about taking pictures. I'll give you the actual solution. So in this case, that is where how we came about to be. We started from overall sequence, the two sequences. And in the process, when we trace back the path, we get your optimal local alignment in this case. And what that particular block shows you for is the alignment till that particular step. And you should repeat this exercise with this scoring mechanism switch to global alignment. Maybe you can also do this as an assignment. And you'll see that we get a slightly different alignment as compared to this, right? Only one single place is where you incurred an indel, which is when you took this path, but everywhere else you're able to align them up to a good enough base. This will change slightly if you change the scoring mechanism. This will also work only if you have, this is also a function of the scoring mechanism. For example, if I had made this +1 or half, it will change by some extent. And where do you think this comes from? Like what score to give? Where should that come from? This, I gave you random numbers, right? But is it totally random, or is it there's something, some ground in what we are doing, or what? Any questions? Anything that was not clear here? Happy to do it again if anything was not clear, but make sure you go ahead, go back with this understanding. So local alignment, we started with global alignment. We said that global alignment is great, but if I have a smaller subset of sequences which I care about, then I want them to match as close to the second sequence as possible. In other sense, you're forcing things to align, even if you do a global alignment, you're forcing things to align end by end, whereas what the interest here is, are there certain subsequences of a sequence which can align, and that alignment can give you a better job, or have more sequence similarity as compared to what you would get if you were to do and force them to have a end-to-end alignment. Okay, so that's what this shows you overall. Yeah.
I have a question. Like once we started the alignment, then after that we are penalizing for the gaps, right?
Yeah.
So then, like, why is it still the function the max of 0?
Why is it?
Like, I thought it was that if there are initial gaps in both of them, that's fine. Or even if once you start the alignment, after that the gaps are penalized, right?
Yeah.
So then why in the function we take a max 0?
So the reason to take that max function to be 0 is because any subsequent steps, if you incur a negative score at each point, you know that that path has already become a suboptimal path. So there is no further point taking or penalizing it or going towards that particular path, right? So if I have a score of -3 or a -5 or a -7 in particular case, in incurring from an indel position, the reason I don't want to see a negative score here is because in some sense it has already become suboptimal. So I'm saying that that is okay, I will discount it and I'm just going to upper cap my negative scores to be 0, so that any other path, so for example, let's say here, right? Where is the best way to show this? For example, in this case, if I knew that these are, this is let's say -2, then I know for a reason that the path till here is kind of suboptimal because I have incurred a negative or a opening gap penalty in this process. The way to relax this is to say that the maximum score that you can get, you're not penalizing negative scores. You're not penalizing initial gaps or opening gaps. Where you're penalizing you're not necessarily penalizing the opening gaps because this is what is set it to 0. And wherever you incur a gap, you're also not penalizing it because we don't know where the alignment should actually begin, right? So any path that you take till that step where you're incurring a negative score should not have a negative penalty, right? So that is a very sort of very simplistic explanation for why we start with incurring a gap of 0, or why we only allow a maximum score of 0. But the other way to think about this is the moment you take a negative path, you have already started your journey towards taking paths where you're going to incur gaps, which you don't actually want to see. You want to see smaller subsequences where you see more and more alignment in some case, right? So that's why you're discouraging any sort of negative score from there. This negative score will have a bleed through effect, right? So if you have a -2, any other deletion that you take from here with that kind of scoring mechanism will always remain a negative score. But that journey is not worth taking. So that's why once I started seeing zeros, I don't have to go through that path at all. That's why I didn't even feel like finishing it. So in this case, I'm already telling you that I know that once you incur a negative here, the rest of the stuff is going to get even more negative. So I upper cap it to 0 and say that, okay, that's a suboptimal path, I'm not going to take it any further.
Is there a possibility that you won't be able to backtrack a path without encountering a zero for the
Is there a possibility that you can backtrack a path without?
You won't be able to find a path without finding a zero.
Without finding a 0? As in, like, so can you give an example? In this case, let's say.
Right. In this case, let's say 2 or 4 was not there.
This was 0, okay? That means that my alignment has not begun yet, right? So that is your indication that if you are coming all the way from here, your alignment has not even actually kind of begin in that sense, right? So while you see these sequences here, right? The reason these sequences are there is because I'm just putting them together, but your alignment actually starts. So I should highlight that. That's a good point. So the alignment actually starts only once you incur a positive score here, right? So that's where it began when you got a 0. The rest of the stuff does not matter, right? So this is where it began. So I should actually have cut it down here. The rest of the stuff you will of course write it, but this is the path that you're going to take because your goal is to maximize that alignment path, right? So it's a subset of the entire sequence in that sense, right?
Right, but given that it's a very long path, can we instead find 2 subsequences?
I see. Oh, can you? Okay. Sure. Yes. Yeah, I see. You can find subsequences which will give you an equivalent score in the alignment. Yes, that is possible. There are 2 possible paths or multiple possible paths depending on what are your sequences and what are your scoring functions, which can give you slightly different alignment. I think we do this in the assignment, so maybe we will have a question. I think I do it in the hands-on. I forget one of those scenarios. But 2 possible alignments are possible actually. Actually, we do this in the assignment, now I recall. So, depending on how you backtrack, there are 2 possible paths. This can happen by the way in Google alignment as well. It's possible, right? So that what and why example that we did, there were 2 possibilities. WH80 and WHY, or WH80 and WH space Y, or WHY space. So similar thing can happen in this case as well.
So while backtracking, there are like zeros in the backtracking into matching,
You don't care about the zeros in this case because all you care about is at what point do your non-zero values start and begin basically.
So but suppose it's like -2, 2, 4, 0, 0, 6, something like that.
0, 0, 6. So 6 is where it started.
So 6 is the second alignment and before 0 is the first
Correct. Yes, that's correct. Yes, so that's possible. Yes, correct. So that's possible. That's a good point, right? So even with one sequences, it is possible to have two paths. I think maybe that was also your question, where you're getting two possible your subsequence, right? So you are aligning your. Maybe let's take an example. So A T G C A T E. Maybe I have T G C. And let me change this to PTT and AC. So in this case, my local alignment will map this and map this. Yeah, that's what. Sorry, that was your question.
So in between will be zeros like.
Yeah, the readings will be zeros there. Right? So that is what it. Sorry, that's. I think I should probably put up a better example to highlight that. But that's correct. So we'll be able to fix that. It all depends on, however, this all comes with this caveat as to what is your scoring function. So in this case, you will have really high score for matches, and that's why we gave slightly higher score actually +2 to enforce that this kind of stuff is better than the rest of the stuff. Okay. Any other questions?
In this one, we are saying that we are encouraging the gaps, right? So, hypothetically, if we swap the scores.
I'm encouraging the gaps in the sense that I'm saying that it's okay, don't worry about it. Like, as in, if you incur them before and after, if there is if it is helping you find out a subset of sequences which can align to the best possible way, then it's okay.
So, hypothetically, if we swap the scores of -2 indel to -1 indel and -2 mismatch, then
Okay. Yeah.
what will be the scenario?
Yeah. Good question. So you're saying this will become -1.
Yeah, and mismatch.
It won't work. I think it won't work. You will be forced to, your indels are suffering less penalty, right? So you'll be a lot more gaps to begin with. So if you, again, it's very going to be, like, there's no way I can give you a generic answer because it depends on what sequence content you have. Because what this is telling you, right? Intuitively, what this is telling you is, indels are okay, but if you have a mismatch, don't try, right? So if you don't want to try, then something like this, for example, G or T, which if I had, let's say, G, C, A, for example, I would say that it's better to, even this kind of alignment is not going to work. So I would just have a very small alignment like CA. Whereas if this had a G here, I'm going to discourage with, actually in this case it is fine, it will still work. If you have a mismatch, okay, let's do an example. So G, B, C, A, and G, C, A. So if you increase the mismatch penalty, then this alignment becomes less useful, okay? Or it won't come. But if you increase the indel penalty, right, which means that I will have lesser chances of having, initially I can have anything, right, like this does not matter. But if I increase my indel penalty a lot, then this is going to be forced to become true. Because indels are very hard, whereas the other alternate case would be this alignment, which could be favored as well in local alignment case. So this and a combination of what sequences you have and what scoring function you have actually tells you what kind of alignment, what kind of alignments are going to be favored. But this ratio will essentially tell you like what is favored. So if you have a indel, in this case indels are favored, right? So if indels are favored and mismatches are not, I will get this alignment. Whereas maybe biologically these are more possible because it seems, if I told you that mismatches are more real in biology, which I will just talk about in just a bit, then I would prefer an alignment like this over an alignment like this. So if I told you that G to T is fine, then I would prefer this alignment over this alignment.
Accordingly, you can check this for you.
Yes, so that is exactly what my question was, right? So quite the way the scoring matrices are figured out is, while we took the very simplest case because that's easy to do in class, but you can have a custom scoring mechanism which tells you that the whether A is getting substituted to G is different from C. For example, what we know in biology and in reality is transitions which are A to G or T to C are more common as compared to transversions, which are any other possible combinations that you can think of. This is purine to purine or pyrimidine to pyrimidine. Again, biochemistry here does not matter, but A to G or C to T is more common. So if A to G is more common, the match, of course, penalty is 2. A to C is -1. A to G is, however, 1. Why? Because I know in biology this is true, whereas this is discouraged because I know it's not very common. Similarly, A to T is not very common, so that's why -1. So based on what we know in biology, this is where, like, algorithm will do its own job, but this information is something that comes from biological information. So that is taken into account while you are inputting or doing any sort of these alignments. Okay. So this is a very commonly used matrix based on this idea that A to G or C to T are more common and other ones are not. So of course, if it's a match, it's a score of 2. But if it is a mismatch, whether it's A to C or A to T, in this case, for example, has higher penalty, whereas A to G is okay, right? So I'm going to discourage. In this case, like if I had an alignment, I would discourage A and C to align, whereas I'm okaying A and G alignment. So that also determines what kind of alignment will you get. Okay. For example, if we align entire human and entire DNA sequence of mouse, we know we can do this because we have access to those sequences. And the actual scores, this is taken from an actual paper, they gave is A to A is 91. It could be any positive number. And A to C is -114, A to G is -31, and A to T is -123. This is just the relative frequency of how common, once we align the DNA sequences, how common do we see A to align to C or A to align to G. So A to C is less common, A to G is more common, and A to T is also less common. This is taken from actual real data. Okay?
Generally, A and T matter, but how are you getting to the other ones?
No, so this A and T is not the complementary part of it. What happens is we have a DNA repair mechanism, right? So our DNA will have certain, at some point it will have certain mutation rate. What has been characteristically understood is it is more common that if you have a A, it will get replaced by a G as compared to by a C or a D. This has nothing to do with complementary sequences. This is only that if you have a single strand of DNA, the repair machinery which is doing its job, it will, if you encounter, let's say, an A sequence, so it has to repeat A, rather than repeating A, it can sometimes make an error, but when it makes an error also, then it is more common for it to replace it by a G rather than by a C or a D. In other words, this is the simplistic version. In other words, when evolution happens, right, so as we are undergoing changes in our bodies and so are all other organisms, the way mutation happens is, while that is random, but the substitution itself is not random. So A has more chance of being replaced by A, of course, but also A can get replaced more often by G as compared to what by C or D in real life, in organisms as well. So that is where it comes from. Okay. Anyone here has heard about Blossom 62? Okay. So the idea behind Blossom 62, this is the first and the only thing I learned when I took computational biology here in this institute, is there are two. While we were doing this at the DNA sequence level, of course, this all can be applied at the amino acid level as well. And in amino acids, if you look at all different organisms and you take their protein sequences, and you take protein sequences which are less, whose sequence identity, which means that the total similarity that they have with, if I take my human amino acid sequence and I take the mouse or horse or cat or whatever, some other organism. I take all such sequences such that any two sequences have less than 62% similarity, and I stack them. I can calculate how common is it for amino acid C to be replaced by S or any other 19 amino acids. Get a score which is called a Blossom 62 score. This is what actually, when I was running the BLAST, this is one of the matrices that is used for calculating what penalty should I suffer if I get a mismatch. So for C, which is one of the amino acids, the score for that is 9. But if its C gets replaced by an S, then it suffers a penalty of -1. It is 0 if it is A, and if it is again K, then it is -3, right? So this has again be something which we have learned from biology. What this tells you is your null hypothesis would be that this should all be equal, but in reality, in real life, we see that the rate of substitution of C by S, K, or any other amino acid is not equal. So this is what is the matrix which is used for alignment in a lot of cases, if you're aligning amino acids. These are called substitution matrices because you're substituting one amino acid for other, and it's called Blossom 62. 62 because that's the percent similarity of the amino acid sequences which were used for, in a sense, training this matrix or training this data. Okay, so I'll just show you a quick example of what does local alignment look like in real life. So this is and hopefully this, I should have probably begun with this question, but I think it's very hard to do it in the class. But what I'm showing you here are two sequences, one from human and one from chicken. And on the left is the global alignment, and on the right is the local alignment. And I will show you the next half. It's a very long gene. It's the HBB gene, which is the hemoglobin gene, which we have both. And of course, there are differences, of course, but one thing that you can kind of figure out from what local alignment is trying to do is, even in cases where it would have led to a long insertion in the case of global, local is trying to find out very small stretches where it can try to find out what is similar, right? So this is that small stretch, which otherwise you will find very missing in the global alignment. So in a sense, the idea of local alignment is, when you find out these very small stretches of sequences where you get more and more identity as compared to what you would get in global alignment. Is that difference clear? And the simple way, right, the reason I asked this question in the last class is the only change that you're doing in the algorithm is those zeros. That's all. And that is creating this magic. Okay, so while we only covered two sequences, in aligning two sequences, you can also align multiple sequences. And this is an example of what's called MSA, multiple sequence alignment. And I usually use this in highlighting in the class for showing you something which I'm currently not able to find myself, but it's somewhere there in the sequences. So let me go now. I cut off the important part. But this is one of those proteins which has, so your 26 letters in the English alphabet, 20 of those are amino acids. And if you're lucky, you will find your name in some protein or the other. So while growing up, we had this term called ego search, which would mean that you search yourself on Google or Yahoo, or if you have heard about that company, and you will find yourself, and the way earlier the algorithms were written is, more frequently you click on those links, they become more and more common. Actually, I found it now. So, this again, you'll do this as an exercise in the class, and that's why this is not about me, but this is about interesting things that you find in biology, right? So, if I focus, then you can find part of your name in the protein sequences. And the reason I used this example was when I started my second half of my research journey, this was the first gene that I was given that I had to find. because it has a very special meaning in the brain. It carries out, brain carries out two general things. One is excitatory and one is inhibitory. So it has an exciting potential, it also has an inhibiting potential. And this is one of the genes that induces that inhibiting potential. And this was missing in a particular species, which is less steady, which is ferret, which is also missing from here, for example. And this figure, when I drew this figure, the reason to draw this figure was to show that the protein sequence has not actually changed, right? So this is a multiple sequence alignment, where you can see that how close, right? Like how close are each sequences. We start from armadillo, elephant, cat, mouse, pig, bonobo, human, chimp, gorilla, macaque, marmoset, turtle, and lizard. And their individual amino acids, like they're extremely constrained. So if something changes, it likely shows that something has gone wrong, right? Or there is something innovative about it. Right? Just look at the colors here. The colors are for each amino acid. You clearly see like a lot of patterns emerging. All the way from cat to turtle, lizard, amphibians, same protein sequence. Very, very similar protein sequence. Right? And that's the beauty of biology. We still don't understand it, however. So this is how you do a multiple sequence alignment, and that can actually be very, very informative to tell you whether what you're seeing, how real is it? Like if it is again there across all the organisms, it definitely has a meaning. And if it is missing from certain organisms, for example, ferret, here, this particular sequence is missing. This is missing not for biological reasons, but for technical reasons, because whoever assembled the genome of ferret did not actually do a good job. But that problem is fixed now. Okay. So if you have to do multiple sequence alignment, this is multiple because this is 2 above more than 2. How would you go about it? Any ideas? We covered aligning 2 sequences, which is called a pairwise alignment. How would you extend this to do multiple sequence alignment? Any ideas? Sorry. If you have to align multiple sequences, how would you go about it? 2 itself is so hard, right? Like putting up such matrices and so on. This looks hard on the board, but you can write a program for this and it becomes very quick. But how would you do multiple? Yeah.
You can do the same thing in 3D, right? It's like a 2D array, just try to.
What if it is a 10D array?
Right, but the computer can do it. I don't think you can manually do it.
Okay, computer, how would you do it in computer? Yeah. How would you use computer to do it? You would create a tensor of dimension 100. Any other ideas? Simpler ideas are always good.
We can scale these penalties, like if 1 minute or 2 minutes, then.
You can scale it, but you still need to take into account so many sequences. If there are 10 sequences, I need to check so many sequences. Any other ideas?
Each of these we align to one which we consider as reference.
Okay, that's a good idea. So you could make one of them as a reference and align each of them, because we know we have algorithms to do that, and we know we can handle pairwise alignment, and that's kind of what is actually done. But before we do that, we just at the scale of things, that why are we doing this, right? Because for this sequence, there were 3 ways to align it. Either you get a match, you get a mismatch, or you get you get a mismatch, or you get an indel in one or the indel in the other. So there are 3 possible ways, which is 2 to the power 2 minus 1. If there are 3 sequences, it becomes combinatorial. 2 to the power 3 minus 1 possible ways. If it is 10 sequences, there are 2 to the power 10 minus 1. So that scoring function itself is going to blow up, right? So one way to do this, one way to handle this is both the complexity and the storage is going to blow up. One way to do this is to do some heuristics, which is you do pairwise alignment and choose 2. You can choose any 2 sequences and do those many alignments, and try to estimate a relationship between these sequences as a guide. So if I have human and if I have mouse, and if I have, let's say, human and chicken and human and frog, then maybe I can just align all of them to human first. So I get one level of alignment for each species, and then try to put it together. That how close is it? And that is what a lot of algorithms that exist out there. One of them is called cluster omega that tries to do this in the real setting, and it works nicely. Okay, so we talked about the Columbus and the egg problem, right, which we have kind of already solved. We will not do MSA even in more detail because at the end of the day it's just a heuristic, so it works, and the central idea is not very different. And this is a story which I've already told, so I'll not repeat. But the thing that you should remember from, or at least I got from this Smith-Waterman algorithm. By the way, sorry, I did not mention the name. The local alignment is called the Smith-Waterman algorithm for two people who discovered or invented, whatever. And the global alignment is called Needleman and Wunsch. Again, two people who discovered or invented global alignment. Smith-Waterman is a very simple extension, right? All you need is this. That's all. And that became a paper. And that paper is one of the very well cited papers in the field of computational or molecular biology. This is Needleman and Wunsch paper from 1970. 16,000 citations. This is Smith-Waterman, 11 years later, 81. 14,500 citations, lesser citations. And then, building upon all this, came this idea of what I showed you in the beginning of the class, which is BLAST, 113,000 citations. So these are citations as to how many people have cited your work. It's a very big number for any field. And this algorithm has done wonders and changed the way we think about molecular biology. So we'll look into that very, very quickly. We do have time to finish it. So the biological problem that BLAST tries to solve is the biological problem that I motivated in the beginning. Is you have found out a sequence in a new organism which nobody has studied so far, and you are interested in finding out what putative function does this particular sequence carry out. You have a database. You have a database of genome sequences of all other organisms. So NCBI, the website that I showed you, for example, has how many 100,000 plus genome sequences of different organisms in one version or the other. Now, if I want to find out where does this small sequence that I have, where does it lie? It's a needle in a haystack problem, because it's a small sequence and it can match to anything. It can match to anything in the world that exists out there. So if I start doing my alignment, this will blow up, right? But if there is a black box mechanism which can tell me that, okay, this is your sequence and it appears in like 5 different species where somebody has already characterized this to have this function, then I have a stronghold that I know that, okay, this sequence is going to carry out this function, right? So that's what the goal is. So we can search, we can compare the sequence with the ones which we have in database, and the underlying hypothesis there is that this is my query sequence which I'm querying. This query sequence will also have some similar function as the reference sequence if I'm able to find out what that reference sequence is. So just to give you the scale of the problem, your DNA is 3 billion base pairs. Imagine you have a query sequence of 500 base pair. Maybe you got some 500 base pair from a new patient, let's say, or new small organism that you're growing in your lab. You want to find out what is its function. So if you do an alignment, its time complexity is O(n) squared, because we didn't fully cover this in the class, but also it's not very hard to understand that if you have to do an alignment, it will, if of length m and n, it will take time complexity mn. Everyone understands what is time complexity? Right. Okay. Time complexity is basically any number of operations that you need to do to be able to solve this problem. Right? But if we have more genomes, if you have 100,000 genomes, if you start doing this number is going to blow up. This will take O to the power gmn or O gmn time complexity. Can you think about ideas to reduce it? So imagine you have this sequence, you have so many long DNAs from everywhere, and you're trying to find out where did it come from. Did it come from here? Did it come from here? Did it come from here? So I have to keep scanning everything from here to here, here to here, here to here. Because I don't know where the start is. I don't know where to begin also. Can you reduce the search space? Problem is clear? So you have a long, very, very long base pair sequence, and you have, let's say, 1,000 plus of them. And you have a very small sequence, like it's like a 5 base pair sequence. I want to find out where did it come from. Somebody gave it to me, I don't find out where did it come from. It could have come from any 5 base pair sequence in this large number of bases, right? And there are 100,000s of them. How can I reduce? Where should I search? Very large search space right now. So how can I reduce it? Any ideas? Any ideas are welcome. From biology, now it's a computer science problem in some sense, but also computer science is also just like any other field, is just logic. So what logic will you apply so that you reduce your search space? You don't want to waste time looking at places where you know this cannot happen. It's as simple as that. How to go about it? No? Nobody? Any ideas?
You can do it in smaller chunks rather than complete it to the button.
Okay, so smaller chunks. So fine, you can take some set of sequences from the longer reference sequences, right? Is that what you were saying? How do you do it?
Only randomly chopping off the sequence and
Okay, so let me put it the other way around, right? So you have a word, let's say for. You have a long report in your Google Doc, and if I to do Control F for, should I start? What should the algorithm do? Should it start searching from the beginning of the report, or can it do something smart at the back end so that any query search that you try to do, it will be very, very quick? So you all do Google search or any search, right? Like for example, I can search for whatever, some function I can search here, right? Like AST, whatever. It's not going to give me anything right now. But once I type, okay, let's do this experiment. So okay, I search for IM. Okay. Now, if I search for IMG or IMA, let's say, should it start its search all the way from beginning, or should it do something smarter?
Start from IM, which is already searched.
Right. So you know it's as simple as that, right? If I know that my search space can be much smaller, if I have an idea about where should these things come from, that is very similar to the idea that is used also in BLAST to reduce the search space. So this algorithm, which is called BLAST, it's a heuristic. Which works on the similar idea that you have a query sequence and you have, sorry, I want to hide this, but it's fine. So you have a query sequence and you have a target sequences. You want to find out where this your query sequences. This is your query sequence, for example. Where can it occur? So even before you have access to this database already, right? Like, but what you don't know is what query sequence I'm going to enter. What I can, however, do is I can do a smart base indexing, or I can prepare my database backend so that any input that comes, I should be able to look them up very, very quickly in what's called O1 time, which is very quick lookup, a hash table based lookup. The way this is done is you take chunks of your query sequence. So if I input this GTCA, I can, or if, let's say this is this longer sequence, I can first figure out for the first 5 bases or whatever, k equal to some x bases, you can try to match them with the database with some heuristic function. What is that heuristic function? Well, the heuristic function is that if I take the first k bases and if I try to score them against all my database that I have, can I get something which is at least has a score of 4 or a 3? That means that these 4 or 3 bases from this 5 bases, that I figured out, they match exactly. So if I take, for example, k equal to 5, I'm going to create a neighborhood. So this is the 5 base pair sequence taken from your query sequence. I'm chunking it down. I'm taking a k-mer of size 5. This is called a k-mer. So it's size of k and mer because it's a sequence mer, right? So it's k. I don't know how to explain it, but it's k-mer, k-m-e-r. Okay, it's like a sequence of length k, which is called a k-mer. For a k-mer, you can have different possibilities. One is, of course, your k-mer will match exactly, but you allow for some mismatches. So you can say that, okay, if I started from GC, ATC, the other possibilities that it can match is ACT, ACTC. You can allow for all other 3 possibilities in each of these locations. And that way you're going to carry out 16 exact matches are possible, right? So what did we do? We took our query, we break it down, we say that, let me first start by matching the k-mers. The way you are matching these k-mers is, first you're saying that if this k-mer is a match, then if I take this k-mer, it should be able to match to a particular neighborhood. That neighborhood is defined by substituting each base with its corresponding bases, right? So in total, you can make, for example, in this sequences, you will make 48 plus 16 matches. The 16 number comes because you allow for for 3 different mismatches at each location. You will allow only exact matches in this case, right? So you're allowing that if I allow for a sequence like CATCG, then at what location in my database sequence, in what location on my query sequence is that k-mer coming from? And I can then quickly do this search. So doing a quick search of a full-on exact alignment is very fast. The reason this was not fast is because we are doing base-to-base alignment. But the moment you fix this k-mer, this is like just doing a grep or a simple word search, right? So this becomes, you can binarize this, you can consider this as a string, and this lookup in a long string can be very, very fast. So that's how the BLAST alignment works. It takes a k-mer, it takes a length of your query sequence, it converts it into a fixed size k-mer sequence, takes your entire database, treats this as a word and tries to find exact match. Exact match is very, very fast. It's not doing alignment at all. So it's just trying to figure out at what positions in your database sequence do you see occurrences of this particular word. Is that part clear?
Also, explain that changing base from part.
Sure. So essentially, when you're doing this sequence merge, right, you want to, so let's say if I take this particular sequence, this has become one word right now, right? I will take this as one word, but when I'm taking it, I also allow for some mismatch.
So 3 different words corresponding to this word.
3 different words as in 3 different bases corresponding to each base. G is already part of it. The other possibility is A, C, A, T. And this to allow for all possible bases, positions, right? So if you do this, right, so there are 3 into 3 into 3 into 3 possibilities, right? If you take all of them, the search that you can do with that particular sequence with your target sequence is very fast. Because you're not doing alignment. You're doing a simple match. That does it match anywhere in my genome? And that search itself is very, very fast. This helps you cut down the search space. So it will tell me that if I take a k-mer of size 5, then at how many, where are the likely starting locations of this k-mer in the database? Okay, that tells me, that reduces my search space. So it's like, this is what I think you had mentioned, Nakul had mentioned in the first class, which is this idea of using, this is called the idea of seeding. So somewhere in, if I take for example this sequence, I do a match and I find out the location of this match as well, which is going to be very quick, again because you're not doing base-to-base alignment. This becomes my seed. Once I figure out that, okay, even though my query sequence is much longer, but the first box here happens to be present at certain location in my genome, I go there directly and then start my base-to-base alignment. And that's what BLAST does, and that's what it is that's why it makes it very, very fast, because it's cutting down time trying to do alignment. It doesn't start the alignment until it has figured out, no, I know that for a reason, that with a very high confidence it has to start here. Okay? So it has cut down the search space overall. Does that idea make sense overall?
That's a simple rapid where it just try to find the smaller sequence matching, and then after that.
Yes, exactly. So that k-mer size is fixed, like you know, usually 7, 9, 11. That is something which you can also decide, but by default it's 5 or 7 in most cases. So it will take a size of k equal to 7, go and find out where exactly does that k-mer will match to a certain extent, and start your local alignment only after that, once it has matched to exactly matched. So how you're saving space is by doing this exact match, which otherwise you would have to first figure out, if you start doing global alignment, you're not doing global alignment or local alignment at all yet, unless you figure out where should I start from. So this step, heuristic, helps you figure out at what point you should start doing this. Okay, so I think we are over time, but very quickly, that's what the idea behind. Is that idea clear? We'll not do a very formal proof of.
If it has something like you are only matching schema of two cells.
Sure.
That is it.
Yes. So usually with small sequences. So okay, good question. Okay, good question. Let's see what happens. So if it is a very small k-mer, right, then things go slightly weird, right? So it's not optimized actually for smart sequences. You have to change the k-mer parameters. So initially I cheated. I did Saket S, for example, and I did a BLAST and it worked. Let me show you in the new window while this runs. I'm going to cut this down by just one base pair. No results. While you can see here, of course there are results. Sorry. You know, this tells me there's like exactly a match of Saket. So by definition there should be a match for Saket, right? But because it's optimized for 5, 7 and so on, like it's optimized for a slightly higher k-mer, it fails. Unless you change the default parameters, it will not find the smallest one. Which should be an easier problem to solve, by the way, right? Like it should be much easier to find out a sort of string of this. But since the parameter, it's not written for small sequences, like it was written with this thing in mind that you have an entire new gene that you have cloned and you want to find out what it is its function. You want to do this very, very quickly, so you figure out likely this is where its starting point is, and then start doing the Smith Waterman as well. Okay? Okay, so we will stop here. Just to review, well, the only thing that I skipped is BLAST score, how this score is done, which we will do in the next class. Sorry, this is not the PDF. So we'll do the sequence, the alignment in the second, the scoring in the second part. Next Monday there's no class, as in 18th there's no class. We will hopefully meet all of you in the guest lecture. The assignment is due on Wednesday. If you have questions, please use Piazza to ask them. I usually discuss the problems in the class before it's due, but I think we will not be able to do it this time because of we're missing the Monday class. But I'm happy to do it on Piazza.